balance for patient. And this change in goal has also fundamentally shifted the paradigm of dose finding design. So in the conventional paradigm, more is better. So therefore, as long as the current dose is safe, and no matter what kind of design you use, 3+3 or model-based CRM or model-assist design, the decision is going to be escalate dose because you want to reach as high as you can. But under the Project Optimus, when our goal is to identify the OBD, we cannot do that, right? Because we're trying to identify the best one, balance risk and the benefit. So therefore, from design perspective, we're going to change our decision to compare multiple doses based on risk and the benefit, and then eventually identify the best one. And this, as a consequence, and this multiple dose comparison will require a larger sample size and also different decision rule for patient assignment. So then the question becomes, how can we achieve this with a smaller list sample size and fast timeline? And this is especially important for industry. And FDA guidance discussed some potential approach can be address this challenge. And two strategy discussed in the guidance is backfill and randomization. And that's the focus of today's talk. And I will first discuss how can we do backfill in a more principled and rigorous way to ensure patient safety and also improve trial efficiency. Then I will talk about how can we incorporate a randomization into backfill. And the ultimate goal is to reduce sample size and improve trial efficiency. Okay, so we start with backfill. And backfill defined as a concurrently assigned patient to doses that have been previously cleared for safety by dose escalation design. So in other words, it's a way we treat an additional patient during dose escalation. And this is distinct from conventional dose or cohort expansion, which typically is done after we finish dose escalation. We identify the MTD or other recommended dose, and then we put more patient on it. And backfill in here, we refers to we treat additional patient during the dose escalation, okay? And there's some benefit of a backfill. And for example, it provides additional data to better inform the OBD because we treat additional dose concurrently in multiple dose. And that's also facilitate multi-dose comparison and identification of OBD. And it also expedited the development because we can collect more data in the same period of time. And from patient perspective, it also improve patient access to the new drug, to the new trial. And a lot of times, after backfill and the dose escalation, we still have a limited data on multiple dose. And in these cases, we may treat more patients, collect more data to facilitate final OBD or recommended phase 2 dose selection. And in this case, a lot of time, we will take a two-stage approach. And the first stage is focus on dose escalation with backfill. And after the stage 1, based on the totality of data, safety, efficacy, PK/PD, and the tolerability. And we will select 2 or more doses into second stage for randomization. And the goal is we want to collect more data and to understand a few doses and eventually to select the optimal dose for subsequent trials. Okay. And backfill itself, actually, it's not new. And in clinical practice and the clinical trial, it has been used for many years. The only difference is before it's done in a very ad hoc way. A lot of time we just treat a few patients. And So therefore, there is a lot of issue never addressed rigorously in the trial protocol, especially given the guidance. People may be more interested to backfill patient to collect more data to satisfy a regulatory requirement. So then the question becomes, under what condition backfill will be commenced and stop? And more important, when you do backfill, you collect more data. And in the case, excessive toxicity observed during the backfill, and should the dose escalation be adjusted to account for the new emerging toxicity data. From statistical perspective, certainly we should do that, right? But if we look at the protocol, especially a few years ago, I never see any protocol have backfill talk about this issue. They just say, we're gonna backfill patient to a safe dose. But the question is, a dose is safe based on 3 patients, 6 patients later on may turns out to be toxic. So in this case, how you handle it. And the other issue is when you do backfill, what's the impact on the operating characteristic? And in here, I'm going to use Boeing as an example to illustrate principled backfill design. And the basic principle and the design and the statistical methods, it's not totally depend on Boeing, and it can be applied to other design. And for example, the keyboard design or MTPI-2 design. Okay, so since we use Boeing as an example to illustrate this backfill strategy, and here it's a quick review of Boeing. It's a very simple decision tree design. And we just need to compute the DOT rate at the current dose, which is a very simple division, number of patients experienced DOT at the current dose divided by number of patients treated at the current dose. And then we look at that DOT rate. If less or equal than lambda E on the left side, which is the escalation boundary, then we're going to escalate the dose. And on the other hand, if it's greater than lambda D, de-escalation boundary, we're going to de-escalate. Otherwise, we're going to stay at the current dose. And in the whole design, the 2 key parameters is escalation, de-escalation boundary. And the bottom table shows the optimized escalation, de-escalation boundary for some common used target toxicity rate. So for example, if we target 25%, escalation boundary, it's 0.197, de-escalation boundary is 0.298. So this is the design looks like when we have a target DOT rate of 25%, which is often recommended target for by regulatory, by FDA. And that decision tree can also be equivalent present, use this decision table. And because the number of patients is discrete, here I list from 3 patients, 3, 4, 5, and so on. And those are the decision boundary corresponding to this simple rate cutoff. So a lot of time in practice, non-statistician may prefer this table, which may be easier to use. Okay, now we back to backfill. And when we talk about backfill, the first question is, when can we open the dose for backfill during dose escalation? And here is the rule we proposed, which consists of 2 rules. The one is clear for safety, which says the dose, the B, okay, it's lower than the current dose C of dose escalation. So essentially, because it's lower than current dose escalation dose, which means that dose is safe, right?
Yeah.
And the second is at least one response is observed at or below the dose B. And this condition is to ensure patient safety. A dose is safe does not mean we can treat additional, it's ethical to treat additional patient. And if it's not effective, why you treat more patient? And also from a drug development perspective, we are also not interested in such dose. We want the dose which shows activity. And in here, the second condition, we use response. That's it's one possible condition. And if you have a reliable surrogate endpoint, or biomarker or PD measurement, you can also use that. Okay, so once we open a dose for backfill, and the next question, when we're going to close it, right, in light of the emerging data. And here is two conditions going to trigger closing a dose for backfill. The first one is observe the DLT rate based on cumulative data at B. It's greater than the buoyant de-escalation boundary. So if we translate it, it just says the data, additional data shows that dose actually is not safe. It's already higher than the de-escalation boundary. Basically, we should consider stop backfill. And the second condition is to improve the reliability of the first condition, and which says the pool DLT rate across B and B+1 is also greater than lambda D. And the reason have second condition is because for the backfill dose, it must be lower than the current escalation dose, right? And in many cases, we see high toxicity. For example, when you treat more patients at a lower dose, originally have 3 patients at 0 toxicity, and then you put 3 more, and you see 2 DLT. And overall, you will see 2 DLT out of 6 patients. And then it's a very small sample size. And the 2 DLT may be just total due to randomness. It's not that dose, actually it's toxic. And because at the high dose, we are still doing dose escalation. So in this case, how can you make a more reliable estimate and decision? So one way is you just pool the data, high dose and the current backfill dose. When you pool the data, if it's still very toxic, greater than lambda D, and in this case, we have higher confidence. The data we observed at the backfill dose may indicate the current dose actually is toxic. So in this case, we should stop backfill. And one nice thing use this pooling strategy is it's maintained the simplicity of the design. And here in the red text, I provide an example. So let's say right now we backfill at dose level 2, okay? And the overall data, including the previous dose escalation data and the current backfill data, overall we have 5 patients, and we observe 2 DLT. And right now, the dose escalation, it's dose level 3, and the 3 patients treated and 0 toxicity. And in this case, to apply the previous rule, okay? And when you look at only at the backfill dose, the toxicity is 40%. Let's assume if our target toxicity is 25%, as I showed at the beginning, per Boeing rule, the de-escalation boundary is about 30%. So right now, it's already exceeded 30%. So that first condition are satisfied. And right now we want to check the second condition, right? We pool the data, dose level 2 and 1 dose higher, which is 3 patients, 0 DLT. So overall, we're going to have 8 patients. And we just need to look up this column, have 8 patients, and what's the decision. And right now, if you look at it, we have 8 patients, 2 DLT. So it's not exceed the de-escalation boundary. So in this case, it's okay. We won't stop backfill, and we can continue backfill as well as dose escalation. And statistically, we can consider more rigorous or complicated methods like isotonic regression to address to borrow information across doses. And but it showed it does not really improve performance. So in general, we prefer this simple approach and easy to understand and easy to use. Okay, and the other condition a lot of time we used to backfill is put a maximum sample size constraint for backfill. And this is more from practical consideration rather than patient safety or statistical consideration. And typically we can set a cap, and if at a certain dose you backfill to that cap, you can stop backfill. Okay, the other unique challenge during the backfill is that additional data from the backfill may conflict with the data observed in dose escalation. So in this 2x2 table, and I listed the decision according to the data observed at the current dose and the decision according to the data observed at the backfill dose. So as we said, the backfill dose is lower than the current dose, right? That's the condition you can open for backfill. Okay, so the reason we use a decision rather than a data, it's we use this decision as a surrogate for the safety of the data, okay? So how we do it, we use a Bayesian cutoff and to measure the safety of observed data. Again, it's going to be located in the escalation interval. That means it's pretty safe, right? Even lower than the escalation boundary. And the stay means it's about okay, we can accept it. And if we use 25% as target, it will be around 20% to 30%. And the de-escalation means it's greater than 30%. So this is a decision, it's just used to surrogate to measure how toxic the data shows. And again, so this is for the current data. So right now, once you have this, and you can see where is there consistent, where there is a conflict, because we know the current dose is lower than the backfill dose is lower than the current dose. That means in general, we expect the dose level B should be safer than the dose level C. So in other words, those lower diagonal means the conflict. And for example, I put here, and if the current dose data shows escalation, that means the current dose is very safe. But at the backfill dose, it shows stay or de-escalate, which shows the even lower dose is more toxic. That's certainly a conflict because we expect lower dose should be safer than the higher dose. So this is the conflict. And sometimes people have questions in terms of the last this cell, and both are de-escalated. So strictly speaking, those data are not directly conflict with each other, right? It only shows the current dose and the lower dose, both are toxic. There is really no much conflict. So in here, we say conflict is more refers to the backfill dose, the data conflict with previous observed data during dose escalation. And then we label this because we still have to address this issue, right? Because this is very different from our original expectation. When we backfill, we think that dose is safe. Right now, that dose is very toxic. And then we label this is for convenience and use the same method to handle all those conflicting cases. And the general strategy to address this conflict is still through a pooling strategy because we want it simple. Again, you can use isotonic regression, and in general, they have very similar performance. And how we pool it. And here we define a pooled toxicity rate, QK. And essentially, it's a data pooled from the where we have conflict. And this is the B star means the highest backfill dose, which data conflict with the data at the current dose C, okay? And so we pool the where conflict happens at dose up to a dose level K, and we calculate the DLT rate. And this, in particular, if this K equals C, that means we're going to pool the data from the backfill dose all the way up to the dose escalation dose. Okay, once we have that pooled estimate, then in the presence of conflict, and we can use that to guide dose escalation, de-escalation, to modify our decision and incorporate emerging toxic data from the backfill dose. And if this QC, you pull the data, pull the DLT rate from the backfill dose all the way up to your current dose. So if that DLT rate is less or equal lambda E, that means it's very safe. And in this case, we just keep our decision escalate dose. And if it's greater than lambda D, that means once you pull and the data still shows toxicity, very toxic, exceeded the de-escalation boundary. And in this case, we should de-escalate to the highest dose, which deemed safe, which have pooled estimate less than the de-escalation boundary. So this is quite a natural decision. And otherwise, we're going to stay at the current dose. Again, the pooled estimate is use a simple tool to reconcile data conflict. So once we have those rules in place, and the last elements we need is how can we allocate the patient between dose escalation and the backfill. And based on trial objective, a different strategy can be used. And one common used one is I call a prioritized dose escalation. And the reason is for dose escalation, typically we have to stagger between cohort. We treat one cohort, wait the DLT period, and before we can make a decision for next cohort. But for backfill, we don't have to stagger, right? Because it's already previously showed relatively safe. We can, once patient available, we can immediately treat in that dose. So to maximize the information in a fixed period of time, it makes sense we prioritize dose escalation. And if one patient arrives, and for dose escalation, we're still waiting for the current cohort to finish DLT assessment. And in this case, we don't have to turn away that patient. We can immediately put that patient in backfill dose. And so this will maximize our patient enrollment and data collection. But again, it's one possible rule. And if you want to randomize between those two processes, or in many cases, you have multiple dose eligible for backfill, satisfy both safety and activity requirement. And you can also randomize that patient into multiple doses eligible for backfill or a dose escalation dose. So that can be adjusted based on your trial objective. So here is an example to illustrate how we assemble all those rules together and put backfill into a dose escalation process. And consider trial. And the goal is to identify a dose which have a toxicity 25%. If you use point at the escalation boundary, it's be 30%, and escalation boundary is about 20%. Okay, let's use that simple as an example. And here we consider the end stop and end cap. So those are more practical consideration. End cap means the total cap for backfill. So if one dose reach 12 patient, which including both escalation patient and backfill patient, once a dose reach 12 patient, we want to backfill more patient into that dose. And the end stop is means during the process dose escalation, if one dose reach 9 patient, we may think dose finding already kind of converged to the MTD or target dose. In this case, we may want to stop the trial for saving sample size. Okay, now we have 5 dose 1 to 5. And starting from lowest dose, we treat the first cohort size is 3. That's why you will see 3 patient, 1, 2, 3. And the blue color indicate the patient has no DLT, okay? And the green means patient yellow gonna have the DLT. And also in here, when you have backfill, you have 2 process, right? So in here, we use a different shape indicate the patient used for dose escalation. We call dose escalation patient, and also a backfilling patient represented by square. And R indicate response. And recall when a dose eligible for backfill, we require that dose must show certain activity. So here we use response. So that dose must have observe 1 response. Okay, once in those places, we can see the first cohort treat dose level 1. None of them had a DLT. And so we escalate to dose level 2. At dose level 2, one of the patient, patient number 5, we observe response. Okay. And then we escalate to dose level 3. At dose level 3, this error means the DLT period. And after we treat the patient, we're waiting for those patients 7 to 9 finish their DLT assessment so we can decide how can we treat the next cohort. And during this DLT window some additional patient enrolled. And by the rule, and we can immediately put those patients in backfill dose. So right now, the dose level 2, it's lower than the current dose, which is dose level 3, and which is safe. At the same time, we observe a response in this dose. So it satisfies two conditions for backfill. So we put patient at 10, 11, up to 13 in this backfill dose. So after we backfill patient 13, and by this moment, we already finished the DLT assessment for the cohort 3, which is a patient 7 to 9. And in this case, it's still no DLT, and we can make a decision next cohort are going to be treated at dose level 4. So that's why we put the new coming patient, the 14 to 16, to the higher dose level 4. Again, and during the time we wait in the DLT window for this cohort, there's a patient 17, 18 registered, and we put those two patients in backfill dose. So up to this moment, actually 2 doses are qualified for backfill, dose level 2 and dose level 3. Okay, so they're both safe, and also both have had a response. But in here, we favor the higher dose because in general, in many cases, higher dose typically more, at least as efficacious as the lower dose. So in here, we backfill to the dose level 3 rather than backfill dose level 2. But again, depends on your trial, depends on the observer data, and you may backfill to dose level, continue backfill dose level 2, or you can even randomize, additional patient to dose level 2 and 3 for backfill. So it's up to how the trial and the data. Okay, and after patient 18 were backfilled, and the patient 14 to 16, this cohort already completed DLT assessment, and still no DLT. So the next cohort going to treat a higher dose, dose level 5. And so in this dose, we observe 2 DLT, okay? But again, during this DLT, we're waiting for those DLT data available, 2 patients available, those 2 patients, 22, 23, they're going to put in the backfill. Okay, once we finish the, okay, so at this moment, when we backfill those 2 doses, okay, dose level 4, we put a patient at 22, 23, we observe 2 DLT. And then previously, the first 3 patients, no DLT, right? That's why we escalate. But when we treat more patients at dose level 2 or dose level 4, we start to observe DLT. And overall, you will notice we observe 2 DLT out of 5 patients. And the toxicity rate is 40%. That's greater than the Bayesian de-escalation boundary, which is 30%, and our target is 25%. So right now, this is going to trigger the rule, should we stop the backfill? So the first condition already satisfied, right? Based on the local data, it's dose level 4, the DLT rate already 40% exceed Bayesian de-escalation boundary 30%. And next condition we need to check is we're going to pool the data with higher dose. So right now, the higher dose already also have DLT. When you put it together, and obviously you will see this toxicity rate will still exceed 30%, the de-escalation boundary. And in this case, we're gonna stop backfill to this dose level 4, and we're gonna put patient 24 backfill to lower dose because this dose is still safe and qualified for backfill. So that's where we put the 24. And after treating this patient, the patient 19 and the 21 already completed DLT assessment. So right now it's the time we make a decision on how to treat the next cohort. And because this dose is toxic, we need to de-escalate. And also this dose is also quite toxic. So we de-escalate to the dose level 3 for treat the next cohort, 25, 26, 27. And again, during the DLT period, and we put a new patient in backfill dose, which is dose level 2. At this moment, the dose level 3, after the patient 24, and this dose level 3 switched from backfill dose to dose escalation dose. But again, those are just for, for labeling, right? Which dose is to the next cohort and which dose is for backfill. And so this illustrated a process how backfill interplay with dose escalation. Okay, I hope this give you a better idea how this work and how this rule play out in a potential trial. Okay, so next we're gonna talk about another common scenario, right? We already figured out how to do dose escalation with backfill. And if you are lucky enough, right? So for example, you work with rare disease and your data signal is strong enough. When you, let's say you backfill 2 doses and during the process, each dose already have 12 patients. And you have very clear PK, PD, efficacy toxic data to support your dose selection. And then you are done, right? And you can convince regulatory, okay, I have sufficient data to support why I select dose level 2 rather than dose level 3. Even dose level 3 is still below or it's the MTD. But in many cases, even with backfill, and we may get 10, 12 patients, but it may not sufficient to support a final dose selection. And the regulatory may want you provide more data, 20 patients, 30 patients. So in this case, typically after this dose escalation backfill stage, I call stage 1, we will followed by a randomization stage. And we based on the backfill and the dose escalation data, we pick 2 or 3 most promising dose into second stage, and we randomize additional patient to those 2 doses to collect more safety and activity data. Eventually, help us determine the optimal dose. So if we do this, right, and one obvious question people always have is, since I already treat a patient in dose level 2 and dose level 3, especially when we put the backfill, we have quite a bit of patient in those 2 doses. And can I transfer those data into the second stage such that I can minimize the number of patients I should randomize in stage 2? And that's a very practical question. And it can save resource and also speed up a timeline. So if we can, how we do it, right? And in other words, all those patients, no matter it's treated through the dose escalation, which labeled by this black symbol, or those blank label, how do we can transfer those patients counted towards this randomization, combine the data together eventually to support our dose selection. And one key challenge here is the stage 1 data are not randomized, right? But stage 2 are randomized. And if we look at the guidance, the reason it's recommend randomized parallel design is because a statistician is clear randomization going to minimize the bias. And also a guidance talk about a lot of time because we randomize, it's a small randomized trial, maybe just 40 patients randomized to 2 dose arm, 20 each. And still, if we do simple randomization, it will still end up a lot of unbalance. So in the guidance, it is specific talk about we can leverage stratified randomization, put the important prognostic factor under control. Okay, so given that, and how can we address this issue, right? And we want to minimize sample size. We want to recycle those data, but at the same time, we want the data as unbiased as possible to mimic randomization. So the key idea here is we can use covariate adaptive randomization in stage 2 to actively eliminate potential imbalance in prognostic factors among stage 1 non-randomized patient. And the design, I call it BARD, and so it allows us to seamlessly combine and also obtain high quality data. Okay, so how we do this covariate adaptive randomization to achieve this goal? And here I'm gonna use slides to illustrate this basic idea. So let's consider we have two important prognostic factors. One is, let's say, biomarker A, okay? And so it's positive and negative. And we're going to use a red color indicate biomarker positive patient, and the blue indicate biomarker negative patient, okay? And the other important factor we want to control during randomization, it's gender, female and male. So female indicated by triangle and male indicated by square. Okay, so right now we have two prognostic factors. Overall, we kind of have a 4 subpopulation. Right now, stage 1, okay, we do dose escalation, we do backfill, and we decide we're going to choose 2 doses into the stage 2 for randomization. So here denote dose level, dose high and dose low. And those are patients treated at those stage 1 in those 2 doses. And you may say, oh, sometimes in stage 2, I may focus on specific subpopulation, right? I may, change eligibility criteria a little bit, or I can focus on specific indication. Stage 1, it's all comer, and stage 2, I just want to focus on lung cancer patient. That's okay, right? Because you just apply the stage 2 rule to screen the stage 1 data. So in here, the data, essentially, it's the effective data. Let's say you want to focus on lung cancer patient, and then you can go back, look at stage 1, at those high, those low, how many is lung cancer patient you treated. Okay, so those are the patients treated as stage 1 on those 2 doses, obviously eligible patient. Okay, so right now, how can we utilize those data, right? That's the key assumption. At the same time, we want to accounting for the stage 1 it's non-randomized. So when you look at here, you can see those 2 doses, the data, it's not totally balanced in terms of those 2 important biomarkers. So right now, there is a stage 2 patient randomization. We have a new patient, which is a female biomarker positive. And how we randomize this patient, right? We can randomize to high dose, low dose. So the strategy is we're going to randomize this patient such that to minimize imbalance between those 2 doses in terms of covariates. And so right now we have high dose, low dose. And here is 2 prognostic, female, male, and marker positive and negative. And this table shows the number of patients fall in this different category. And so for example, in high dose, we have 5 female. In low dose, we have 1 female. And high dose, we have 5 marker positive patient. And the low dose, we have 2 biomarker positive patient. And the last shows the difference, okay? So the difference for the male, female, this prognostic factor difference is 4 in terms of counts. And the biomarker is 3, and the total difference is 7. Now, if we are going to assign this patient, new patient, into the high dose, and we're going to put 1 more patient in this category, and the total imbalance going to becomes 9. And if we assign this patient to low dose, okay, those 2 cells increase by 1, and the imbalance is 5. So obviously, we want to assign this patient to low dose because it minimizes the imbalance in terms of those prognostic factors. So you can do that in deterministic way. Okay, I always be greedy, directly assign to the dose that minimize the imbalance. And another approach is you skew the randomization. Okay, you still want to keep some randomness because otherwise it's under criticism, you are total deterministic. So in here, for example, you can use a 90% chance assign it to the low dose, 10% chance assign it to the high dose. So if you are familiar with this technique, you will recognize it's basically the idea of Pocock assignment minimization. But the difference here is from a standard Pocock assignment minimization is here you can view this as a conditional Pocock assignment, right? Because before you do covariance randomization, you already have some data, and you condition on those data, trying to minimize imbalance between those arms. And again, the goal is slightly different. So here is we use that mechanism to eliminate the potential imbalance from stage 1, such that at the end, hopefully we will get randomization very similar to pure randomization. And In terms of minimization, if you're familiar with it, you know it's a very widely used randomization strategy, and it's also mentioned in FDA guidance in terms of adaptive design, and it's also acknowledged, you know, this does not directly increase type 1 error probability when analyzed with appropriate methodology. But in here, it's less concern, right? We are not really trying to make a very powered, rigorous comparison after all, as noted in FDA guidance. So you don't have to, you know, rigorous power for the multi-dose comparison. You just need sufficient data and to maximize unbiased comparison. And also a lot of studies shows minimization also have very good performance compared with simple randomization, and shows it can be similar or even better than stratified randomization. Okay, so here I'm gonna, yeah, I think I'm gonna finish in a few minutes. So here I'm gonna show some operating characteristic of this new approach and show how it really performs amazing and very similar to pure randomization. So what we do is we compare this barred Bayesian. So essentially in the first stage, you do Bayesian dose escalation with backfill, and then you are connected by this covariate adaptive randomization. And with Bayesian with simple randomization. So in this simple, you do first you do dose escalation, and then you select 2 doses, and then you randomize patient, do simple randomization to those 2 doses. Okay. And this 5 dose maximum sample size is 30, and the backfill up to 12 patients per dose. And we're gonna select MTD and MTD -1, which identified at the end of stage dose escalation into stage 2. And we target a sample size of 20 per dose arm. Okay. And the target 20 per dose arm means different implication for those 2 design, right? For the first design, because we can recycle the stage 1 patient counts towards stage 2. So that means in the stage 2, we don't really have to enroll additional 20 patients per arm. Okay, it will translate into sample size saving. But for comparison, for this single randomization design, we're going to discard all the dose escalation data. And for the MTD and MTD-1, we enroll additional new 20 patients per dose arm. Okay. And we consider 3 important covariates and prognostic factor. And we're going to assume X1 and X2 are used for adaptive randomization. But in here, we intentionally assume there is another prognostic factor unknown. So we didn't include this into randomization. And we also compare with the pure post-hoc randomization. So this is going to use for gauge how good it's our process, because we mix the randomized, non-randomized. How about compare with pure randomized based on post-hoc assignment from beginning? Okay, so we consider different cases. I want to go to detail. Essentially, for example, scenario 1, you know, the first row shows the DLT rate, its monotonic increase, and the second row is the efficacy of each dose, and the last one it shows utility, which is measure the risk-benefit trade-off. You can see different scenario represent the optimal dose at a different dose level. Okay, so here is results. And the first message is you can see as expected, when you use this barred design, you can save sample size. It's obvious because you recycle the patient from stage 1 and move forward into stage 2. So how about the imbalance, right? You can use the stage 1 data, that's good. The question is, does that compromise the covariance imbalance? So here shows the covariance imbalance comparison. So you can see surprisingly, so here shows the imbalance of first covariance X1. And you can see surprisingly, the bar actually have much better covariance balance than simple randomization. Okay, so it's, and this vertical line indicated the pure post-hoc assignment. That means I just take a new 40 patient, do post-hoc assignment randomization. As I showed at the previous publication, previously shows the post-hoc assignment at least comparable or even better than stratified randomization. So you can even look at this as stratified randomization should be also around this line. Actually, this should be better than stratified randomization. And then you can see through this bar design, we almost reach, kind of optimum covariance balance. Right? So this is amazing because we mixed the stage 1 patient, which are not really balanced or active balanced. Then it's just due to random. They have difference. And this shows how the balance of unknown the covariance. And the take-home message is you can see the bar is very similar to POCO assignment. So that's the best we can do, right? And this shows how can we identify the OBD. And you can see with them reduced sample size. The yellow one, it's the barred point. It yields better OBD identification than the simple approach. You do dose escalation and then followed by randomization. Okay, to facilitate the use of those novel design, and we also developed some app at trydesign.org. I think many of you are familiar. And if we go to our website, it looks like, and if we go to the Bayesian suite, and you will see there's a newly added panel. It's backfill. And in here, you can find the BF Bayesian and the BARD. And so this is essentially the app interface looks like. It's pretty straightforward and easy to use. And here's the last slides. And if you are interested more detail and especially technical detail, they are included in those two publications. Thank you for your attention. I'm open. I'm happy to take questions.
Thank you very much, Dr. Yuan, for the very nice presentation. And yes, actually, in fact, there are a lot of questions on chat. I can read the questions for you maybe. And the first question is from, maybe you can also see by yourself, right?
Yeah. Okay, yeah,
okay.
I can. Yeah.
Okay, great.
Let me switch this. I turn on the laser. Okay, I can see it now. Okay, yes. Okay, there's some recent tendency incorporate later on toxicity happen in DLT into dose escalation decision can backfill principle for the later onset toxicity. They are not coming in different patient as backfill. So we are any problem. Okay, yeah. So that's a good question. So we are working on that, and in principle, intuitively, you think it should not be a problem. And the only challenge is how can you incorporate pending data into all those rules. I think we are very close to finish the manuscript, and our preliminary result shows still performs very well, and especially we compare with those counterpart later onset design with and without a backfill, and it shows the same benefit for the case similar to the case you have no late onset toxicity. So hopefully I can share that pretty soon. Can we always pull? The other question is, can we just always pull after backfill versus only pull in case of conflict, though the results may end up being the same? Yes. I don't see much reason, rationale to pull if there is no conflict, right? And if the data, backfill data, continue support your backfill dose is safe and efficacious, then you just continue backfill, right? You don't have to do any estimation. You don't even have to worry about backfill dose. So yeah, it's only in this conflict you have to worry. The observed data is due to randomness or it's a true safety signal. And you want to make sure. That's why you pull, get a more reliable estimate. And the next question is, if we randomize backfill during escalation, we can end up in a scenario where highest dose can no longer be randomized. Would the health authority accept to choose dose in in this scenario or require another randomized cohort to confirm the dose? I don't quite totally get the question, because when you randomize, and there's two ways you randomize, right? You can randomize among doses qualified eligible for backfill, or you can randomize also including the dose escalation dose. That's basically the mechanism how you assign the patient. And we will never assign the patient beyond the current dose escalation dose. And when the highest dose can no longer be randomized, I'm not quite getting it. So if it's not safe, certainly we cannot treat the patient over there. It's have nothing to do with randomized. Maybe I'm not totally understand the question. Okay, but there's many questions. Let's first go through this. If you need have time, we can go back to that question. The tight CRM tight bone was developed to enable decision making for trials, even when patients are pending DOT assessment. Although they serve different purpose, if you prefer not to delay trial due to pending DOT, could the backfill strategy be considered a better alternative to tight CRM tight bone? So this is a very good point. Yes, I think this is also a potential benefit of backfill. And yeah, I missed it in my slides, so thank you. So this is also a very important benefit of backfill. And during the time you're waiting for DOT window, so when you use backfill, it allows you to accommodate those new patients. In the case of a phase one, next question is, in the case of a phase one study with a broader therapeutic window target dose, could the initial single patient intra-patient titration approach allow for fast dose escalation when risk of toxicity low, accelerate the process funded MTD? The answer is yes, and that's a very widely used practice. And but you need enough data to convince regulatory or no matter it's FDA or IRB, the low dose it's a lot of signal suggests it's very safe, and you want to maximize patient benefit. That's why you want to minimize patient at that low dose. So in many cases, we set a cap on the dose titration. And for example, first two doses, you're going to titrate. And after that, no matter how safe it is, and you're going to switch to 3 patient. And the next question, in this example, it looks like we have to pause enrollment after patient 21 is assessed. Only after assessing 23 do we enroll 24. But with the bone de-escalation, why not enroll into dose level 3 while we're waiting for patient? Let me see. I think that one, I need to go back 2 slides. So let's put this a little bit late. I can answer the other question. If the other question is easier to answer, we can go back to this question. The other question is, the randomization probability should be sensitive to the final operating characteristic. Is there a guidance for pre-specify this randomization probability?
So maybe I can a little bit clarify. Is this from I mean randomization probabilities for the RCT part for the stage 2, because I see that you have the 10% and the 90%, right?
Yeah.
I just yeah.
Yeah, that's a very good question actually. So what we found is how aggressive you want to use that randomization. It depends on several factors. The first is how many patient you need, new patient you need to randomize. And the second is how imbalanced or what's the percentage of data you're gonna carry forward at stage 1. So the reason is, if the second stage you have a relatively large sample size compared with the stage 1, the carry forward patient, that means you have more chance to rebalance those 2 dose arms. So in that case, you don't have to be that aggressive, right? Because you have enough time to balance them. But in the case, for example, in the stage 1, let's say you want to speed up pipeline. You at the stage 1, you do a lot of backfill. And in other words, you're going to have a lot of patients are going to move forward into a stage 2. And in this case, you have a limited number of patients can be used to rebalance the 2-dose arm. And in this case, you should favor more aggressive assignment to eliminate those imbalance. So you can put that into consideration. And how is utility defined? And yeah, because due to the time limitation, I don't specific talk about it. And here it's kind of when you have considered binary endpoint, it's a 2x2 table. You have 4 different outcomes, right? You have toxicity, no toxicity, efficacy, no response, no response. And then you assign a utility score for each outcome and calculate it. So that's one way to do it. And the other is you can also use very simple difference trade-off between the response rate and toxicity rate and so on. And could the slide deck be circled to audience? Yes, I will share after the presentation. Would have those cohort 2 evaluate patient instead of 3 affect Bayesian performance? So in general, the impact is very minimal. And what's more important is number of dose and the number of patients. And remember, the Bayesian is adaptive decision process. And you can think it's a continual learning up and down through each cohort. As long as you have reasonable number of chance to go up and down, learning that dose toxicity relationship, you will end up very similar operating characteristic. So that's you use 2 or 3. Yeah, it doesn't matter too much. Okay, so we are 11. Okay, that's good. I don't see any new question. Let me go back to the question. I needed, in order to see both, I needed to go back to the slides. Okay, that's a question regarding specific question regarding that illustrative backfill example. Okay, so it looks like we have to pause enrollment after patient 21 is assessed, and only after assessing patient 23 do we enroll patient 24. Okay, that's a very good question. Okay, so here is for the when we decide if a backfill dose should be stopped or not, we based on real-time data. Okay, so in other words, we're going to ignore the pending patient. We just based on the data at that time available. So let's say, for example, when we backfill patient 23, and there's 2 cases may happen, right? One is very quickly, within the first week, patient 22, 23 both have DLT. And in this case, we notice it's too toxic. That's why we can immediately, and after 1 week, patient 24 are enrolled, and then we can immediately treat patient 24. Okay. So in other cases, let's say we put the patient 23 at dose level 4, and we don't see any DLT, but we only follow the 1 week. And by that time, patient 24 come. And by that time, the real-time observed data is we're going to have 4 patient data at dose level 4, which is the patient 14, 15, 16, and the patient 22. And for patient 23, this patient have not finished DLT window yet. We won't use that patient data. So if that's the case, you will see we're going to have 1 DLT out of 4 patients. And if by that rule, and still safe, and then we will put the patient 24 still in the dose level 4. Okay, so that's a simple way we address that issue. And people may ask, you still have some pending patient, pending data for 23. Why you totally ignore it? can you use tight Bayesian, tight CRM, those kind of strategy to accounting for that partial information? The question is yes, and we are working on that, and our simulation shows the impact is pretty minor, and you're accounting for this pending patient or not. It doesn't matter too much. But in principle, you will think, why I just based on observed data, right? I should also account for those pending patient. But the interesting is for the backfill purpose, it doesn't matter too much. And in terms of overall OC, it's very, very similar.
Thank you very much. And also maybe one last question, and and we can just end our session today. So the last question is that in this example, why de-escalate from dose level 5 to dose level 3?
Yes, that's a good question. Because the backfill put additional data, right? So essentially, when we finish the DLT assessment for the cohort at dose level 5, 2 out of 3 DLT, and per Bayesian rule, you're going to de-escalate dose level 4. But at dose level 4, you already have data, right? And you typically in the standard Bayesian, without backfill, you don't have to worry. You just de-escalate dose level 4. But right now, because you get additional data, which from patient 20 to 23, so you need to reassess if this dose is safe enough. Obviously not. It's 5 patients, 2 DLT. It still exceed Bayesian de-escalation boundary. That's why you will continue to escalate to dose level 3. And dose level 3, based on the data, including backfill and also previous dose escalation data, it's pretty safe. So that's why you continue put the next cohort to dose level 3, which means you de-escalate twice from dose level 5 direct to dose level 3.
Okay. Thank you very much. I think the time is already passed. And thanks again, Dr. Yuan, to give us this very wonderful presentation. And thanks everybody for participating and your questions. And I think that we can see you on the next our ISBS webinar. Thanks, everyone.
All right, thank you.
Thank you, Dr. Yuan.
Bye-bye.